Hello everybody and welcome you all to this video lecture, that is structure of TMV and Bacteriophage. This video lecture is being presented by D. Rajashekar, lecturer in botany, Srimati Ganganeni Kalyani, Government Degree College, Vinikonda, Guntur district. Here in this slide you can see tobacco mosaic virus, the first discovered plant virus. It is an example of typical plant ribovirus. It causes a disease for tobacco plants, that is tobacco mosaic disease, studied by Mayer because it was a serious disease for the tobacco crops all over the Europe continent. The experiments done by Mayer were repeated by Ivanovsky and proved the existence of new pathogen in the world, which was less in size than bacteria. But the virus was first crystallized by Wendell Stanley in the year 1935 by using the techniques like X-ray diffraction, electrophoresis, ultracentrifugation, negative staining, electron microscopy, and double shadowing. Thus, the structure of TMV became familiar to everybody. TMV is a rod-shaped virus with helical symmetry. As of other viruses, it is a nucleoprotein showing two components in its structure. One is the outer protein coat called capsid and the inner core, that is single-stranded RNA. It is actually 300 angstrom units in length and 180 angstrom units diameter. It is with 95% proteins and 5% RNA. As I told, RNA is a single-stranded one, that is an important feature of any plant virus. RNA consists of 7,300 nucleotides and its molecular weight is approximately 2.3 millions. Capsid is made up of 2,200 capsomeres around the single-strand RNA. The capsomere's molecular weight is 17,400. Each capsomere is made up of 158 amino acids, and these capsomeres show 130 turns around the nucleic acid. Each turn is with 16 capsomeres. And the total molecular weight of TMV is 39 into 10 to the power of 6 Daltons. So it is a plant virus with the protein coat and single-stranded RNA. First identified virus, first crystallized virus. It is a rod-shaped virus with helical symmetry. And here in this slide you can see another virus, that is bacteriophage virus, which infects or attacks bacteria. So it is the smallest virus of all, below the size of bacteria. That is why it can infect bacteria. The structure of bacteriophage was first studied by Twat in 1915 and confirmed by Deherelli in 1917. Actually, T2 and T4 even type of bacteriophages were well studied, and the details we have presently with us are mainly from T-even type of bacteriophages. Bacteriophages show tadpole morphological structure, and the symmetry is binal symmetry. Each phase is having two parts, head and tail. The head of a bacteriophage measures approximately 65 to 95 nanometers, and it is with the cubical or icosahedral symmetry, and the tail is a long structure. It shows actually helical symmetry, and the tail is enclosed by a contractile sheath that is called a tail sheath. In between the head and tail, there is a special structure called collar. The function of a collar is still unknown. In the head region, you can see double-stranded DNA molecule. The DNA of the phage is having 5-hydroxyl methyl cytosine instead of normal cytosine. The phage is with the tail plate or base plate at the base of the tail region, from which 6 long thread-like foldable tail pins or tail fibrils form. They are useful for attachment of the phage to any host, especially bacteria. So bacteriophage is a virus which infects bacteria. They are smallest known viruses, discovered by Twat and Dihrali separately. They are tadpole-shaped structures and with binal symmetry. Head is with cuboidal symmetry and the tail is helical symmetry. It's a mixed type of symmetry. Head is with a double-stranded DNA. Tail is a long structure enclosed by a contractile sheath. Collar is the region that is present between the head and tail. Tail is ended by the base plate or tail plate, from which 6 tail fibers are formed. Thank you for your sound listening. We'll meet in another video lecture. Till then, bye.